Hello, this is Tracy Koch, and I'm going to lecture today on the genetics of cancer in chapter 14 of your textbook. So as we all realize, genetics does play a role in cancer development and treatment, and we know that cancer is a very complicated disease with multifactorial considerations for the disease development. So in this chapter, we're going to do an overview of those changes and some of the considerations when looking at cellular changes with cancer development. It's a good idea to review some of the previous content that we've talked about in our textbook, maybe even going back to review chapters 1, 2, and 3, and 4, just to kind of be comfortable with that content on basic DNA structure, control of cell division, protein synthesis, and Mendelian inheritance. So those will all be concepts that we do cover when we're talking about the genetics of cancer. So we know that all cancer is genetic, but is all cancer inherited? So these are two kind of slippery slope statements. But we can say that positively all cancer is genetic, because the genetic development of cancer involves the changes within your genes that regulate cell division. As far as the statement, all cancer is inherited, it's not really a simple or straightforward yes or no. Cancer is inherited from one cell generation to the next. However, most cancers, or the risk for developing most cancers, is not inherited from one's parents. It is of note, however, that there are some exceptions to that rule. Cancer facts. All cancer is genetic in that it is always caused by changes in the expression of genes involved in cell division. So remember, cell division is a very important part to cancer development. Cancer cells transmit gene changes from one cell generation to another, but cancer is not inherited from one human generation to another. But remember the exception to the rule. Specific mutations in genes regulating cell division that occur in germline cells can be inherited from one human generation to another and greatly increase the risk for cancer development. Important things to remember is that cancer is a disease of cells. It arises from normal cells that have been altered at the DNA level. Even one cancer cell is capable of generating widespread metastatic disease, hence a monoclonal origin of cancer. Cancer develops as a result of three interacting factors: environmental exposure to carcinogens, health of the immune function of the organism, and genetic predisposition. So those are really kind of, I think, the three main concepts when thinking about cancer development that you should keep in mind. So in order to understand a little bit better how cancer cells function, or dysfunction rather, it's good to look at what normal cell function is. So let's kind of review that. Normal cells are orderly. They are well regulated and have well regulated growth. They have a specific morphology. They have a small nuclear to cytoplasmic ratio, differentiated functions. They are tightly adherent. They're non-migratory. They're euploid, which means the cell nucleus has a normal number of diploid cells for that organism, and they respond to signals of apoptosis, apoptosis is your programmed signal, programmed cell death, So characteristics of early embryonic cells are that they are rapid, continuous cell division. They re-enter the cell cycle immediately after mitosis. Anaplastic morphology, so they're all small and round. Larger nucleo to cytoplasmic ratio. No differentiated functions. They're loosely adherent. No cell adhesion molecules, or CAMs. You'll see that abbreviation in the text. They're able to migrate, and they're euploid. They only feature in common with mature differentiated normal cells. A benign tumor cell has a specific morphology, the small nuclear to cytoplasmic ratio, differentiated functions, and tightly adherent, non-migratory, euploid, continuous or inappropriate growth not needed for normal cell function. So this tumor cells grow by expansion and they don't invade. Benign tumor cells, that is. In contrast, when you're looking at the characteristics of cancer cells, they too are rapidly continuous cell division. They re-enter the cell cycle immediately after mitosis. They don't respond to apoptotic signals, so they don't have that automatic shut-off or cell death that you have with regular cells. Genetic regulation of cell division is lost. Anaplastic morphology, large nuclear to cytoplasmic ratio, no differentiated functions, and they are loosely adherent. No CAMs are present. They are able to migrate, and they're usually aneuploid. The anaplastic morphology, progressively the cells lose the parent cell features. They become small and round, and they resemble the tissue of origin, but they may be undifferentiated with characteristics of a cancer. They have a larger nuclear to cytoplasmic ratio. This is a picture of how the cells are loosely adherent. They're able to migrate. Some other cancer cell features is again, the loss of the parent cell differentiated functions. They stop producing the CAMs, and they gain enzymes on the cell surfaces, which lead to more leakage, tumor breakage, and release of cells into your bloodstream. So that's another way that they spread. Tumor cell migration and invasion into other tissues, metastases, which we just mentioned. The gain of metabolic and selection advantages that allow the cells to continue to grow when the host nutrition is poor, and they are all related to genetic or gene alterations. The malignant transformation is the process of the changing of a cell from a normal phenotype to that of a malignant phenotype. This is a stepwise, time-dependent, well-characterized progression. So it is not a chaotic, willy-nilly mass of just chaos. It is a very organized process. It involves cell alterations at the gene level. So this is a graph that depicts the steps of initiation and promotion in transformation of a normal cell to a cancer cell, and allows it to progress to a malignant metastatic state. So we're going to go through this in a little bit more detail through each step. And here's a little bit different way to look at it. The steps of initiation continue to promotion, to transform the normal cell to a cancer cell, and then further promoting into a highly malignant and metastatic state over time. Initiation is the actual damaging of the DNA, either directly or indirectly, by agents or conditions that result in permanent alteration of gene expression. It does not interfere with the altered cell's ability to reproduce. So some common initiating classes of agents are chemicals, physical like radiation, or biological, different viral infections, unrepaired spontaneous DNA replication errors, or inherited DNA mutations. Mutations in genes regulating cell growth lead to the development of cancer. Oncogene products, including cyclins, are the CAR's accelerator, and your suppressor gene products are the driver and the brake. So that's what stops the process. This is a little pictorial analogy that the author of the textbook makes, and I think it does help with kind of getting that picture in your mind of the oncogene being what's revving up and moving, and then the suppressor genes is what pulls back and stops the process, or slows it down at least. So you have oncogene overexpression with a loss or mutation of suppressor genes. Cell growth and proliferation leads to possible cancer. Driver mutation is the clear linkage to cancer development and growth advantage responsible for the cancer phenotype. The passenger mutation is seen within the cancer genome but not linked to growth advantage, so this is really irrelevant to your cancer phenotype. Here are some suppressor gene mutations and associated cancers. This is in your textbook, and you might hear these terms being thrown around medically, and especially if you work in oncology, this is all likely looks familiar to you. but I'll let you review that in the textbook. So let's talk a little bit again about oncogenes and what they are. Oncogenes are normal cells that regulate the cell cycle. It's the code for cellular proteins that relay signals to the nucleus, and the signaling process begins at the cell membrane. So it relays the system of signals through the intermediaries in the cytoplasm, and it ends in the nucleus with an activation of the transcription factors that help move the cell through the growth cycle. So pro-mitotic oncogene products would be various cyclins, aurora kinase, and various cyclin-dependent kinases. So these are products inside that are pro-mitotic. And these are some of the mutations that are associated with cancers. So the suppressor gene products regulate your cell growth. They limit or inhibit the expression of the oncogenes, and they induce apoptosis when needed. And remember, apoptosis is that programmed cell death. And these are examples of suppressor genes. So mutations in the suppressor genes, it can even be just one allele, will reduce the ability of the suppressor gene products to inhibit the activity of the pro-mitotic oncogenes. So even one small change in one allele will affect that suppressor gene's ability to inhibit the activity. Mutations in suppressor gene alleles or DNA repair genes cause most cancer development. Mutations in oncogenes, although occurring less frequently, result in the loss of oncogene sensitivity to suppressor gene regulation. Both types of these mutations lead to oncogenic overexpression. These are your repair gene mutations that are associated with cancer. When talking about oncogene expression, I think one of the easier ways to think about this is just break it down to some basic facts. And if you look at the race car analogy that we saw earlier in the slideshow, if you look at the first car at the top A, let's say both sets of brakes are working front and back. So those brakes work when the driver slows down and uses the brakes, the car will stop well in front of the tree. In example B, let's say the front brakes are broken or they're not working properly, so it does take longer for the car to stop before hitting the trees. He doesn't hit the trees, but he doesn't stop nearly as quickly as car A. And in example C, you have both sets of your brakes are non-functional. So let's say both of your suppressor gene alleles are non-functional. The car cannot stop and hit the trees. So your suppressor gene alleles, when working properly, will stop the progression and changes in the cells that lead to development of malignant cells. Promotion is the enhancement of expression of the gene altered during initiation. Promotion usually increases the growth rate of the cells and is a reversible event. Promoters can be external to the cell or internal, like enzymes, hormones, or natural growth factors. And here we're back to our little promotional mutational timeline. Malignant transformation from a normal cell to cancer cells after the exposure of a mutational event. So it can take anywhere from months to years. But you have this continuous exposure to those promoters. And again, looking at initiation, here's your promotion, promotion, promotion. So this over time exposure to the promotion will transform a normal cell into a cancer cell and then allow it to progress to a highly malignant and metastatic state. Progression is the continuing change over time of a cancer cell population, causing the cells to become more malignant, more aggressive, and less normal in appearance, function, and growth regulation. So progression, you have increased growth rate, increased invasiveness, metastases, biochemical and morphological changes that make the cell appear less normal and function less normally. Metastases, I'm sure, is a term that you've all heard before. This is the special ability of a cancer cell to invade other tissues and organs, forming secondary tumors either by direct extension or by separation from the original tumor, so breaking off. It's most commonly spread via the circulation or the lymphatics. So metastases is a pretty complex process. You'll note that those loss of adhesions of the molecules of the cancer cells, so they can adhere, or so they can break away and be spread and travel through the circulation. So that's a pretty simplistic view. It's actually a really complex process, and many cancers have a predictable pattern of metastases. So it's noteworthy to, some breast cancers metastasize to bone cancer, prostate cancer metastasizes to bone cancer. So those are some things to keep in mind, that pattern of metastases can be predictable for some cancers. So this is some classifications of looking at different cancers, and when you look at a pedigree, maybe classifying them, so to speak, into different ways that you identify them in the pedigree or within a family. So sporadic cancer would be a cancer that occurs as a result of environmental exposure or unknown factors. There is no real observable pattern of inheritance within the kindred. So it's not a clear autosomal dominant pattern or a very clear familial pattern. It's sporadic. The mutations through carcinogenesis disrupt regulation of cell division, usually somatic cells. It's not present in higher than expected levels within 3 family generations. Sporadic breast cancer occurs without any observable pattern of inheritance. It is not present in a family at a higher than expected level, so less than 1 out of 8, and affected individuals are all postmenopausal women. This is a pedigree, typical family history and pedigree showing sporadic breast cancer, and you can see 72-year-old who's diagnosed, but you can see it's not an observable pattern. Familial cancer is a cancer that occurs at a higher than expected frequency within the kindred. It does not demonstrate any observable pattern of inheritance. The family may have a higher than expected incidence of several cancer types, and those who develop the cancer are older, usually over the age of 60. Again, it occurs without any observable pattern of inheritance. It's present in the family at a higher than expected level, greater than 1 out of 8. Families may also have a higher incidence of other types of cancer, and the affected individuals are all postmenopausal women. That's familial breast cancer. And here is a pedigree or a typical family history of showing familial breast cancer. It's a really nice pedigree, and it should be in your textbook as well. But you can see those diagnosed were all older, postmenopausal. There are other types of cancer in the kindred. Hereditary or inherited cancer is cancer that occurs with an observable autosomal dominant pattern within a kindred. Diagnosis is at much younger than expected ages. It's caused by germline mutations in suppressor genes. The risk is inherited, but not the actual cancer. Risk is greatly increased, but not absolute. So just because the risk is there does not mean you're going to get the cancer. Hereditary breast cancer, it's an observable autosomal dominant pattern of inheritance, present in a family at much higher than expected levels. It's often associated with other cancer types. Affected individuals are women and men of any age. It is bilateral disease. And here is another pedigree and family history showing an autosomal dominant pattern of inheritance for inherited cancer, BRCA1 positive individuals. And, so this concludes our lecture on the genetics of cancer. There's a lot of information in here, and hopefully it was presented to you in a way that you understand it, and certainly it would behoove you to review the text chapter 14, and we have a case study for you to for review to help you understand the principles of the different patterns of inheritance. And if you have any questions, please feel free to contact me via course email. Thanks for your attention.